Atif, you're there? Would you like to give me a quick recap of the last class? Atif, am I audible to you? Am I audible to others?
Yes.
Yes, so Mamuna, let me go to you. Would you like to give me a quick recap of the last class? Yes, please.
So we learned that how does male reproductive system function and control? How is it controlled? So we learned that the entire male reproductive system is dependent on hormones, which is hormonal regulation, which consists of FSH and LH. FSH is follicle stimulating hormone, and LH is luteinizing hormone, which is secreted by the pituitary gland. And the hypothalamus helps to balance the hormones secreted by pituitary gland. And then the primary hormones involved in the functioning of male reproductive is the FSH and LH, and it is produced by the anterior pituitary gland, and FSH is necessary for spermatogenesis. LH stimulates production of testosterone, and then testosterone produced by testes under the effect of helps in maturation of sperms. Then hypothalamus. Then we learned castration, which is the removal of testosterone. Then
Castration is the removal of testes, testicles or testes. Testosterone obviously will be removed if there are no testes, right? So
yes.
on castration, the only hormone that the body loses is testosterone. It does not loses LH and FSH. Okay, so this was once asked. Upon castration, which hormone is not present anymore? So testosterone is not present, but LH and FSH is still present because it is produced from pituitary. It is just like it has no organ to go and show its effect on. Okay, so it is there, but it will not do anything. Okay.
Yeah. Then we learned what happens in puberty in males. So we learned that we see facial hairs, we see enlargement of Adam's apple. Then the voice become coarse, and it is also low pitched. Then muscle development, bone mass, pubic hair, fat distribution, and sexual rise.
Yes.
Yeah. Then we learned that about penis. It is used for the sexual intercourse. Then
it is the secondary reproductive structure. I told you that about the primary organ we'll study today.
Yeah.
Yeah.
Okay, so the secondary structure is penis, which is used for sexual intercourse, and the shaft, that is the penile body, it is cylindrical, tube-like structure, and then there is a foreskin at the tip of the penis, that is head of the penis, which helps in the lubrication during the sexual intercourse. And that's all I think.
Yes, right. And the last thing that we were discussing yesterday was this sexual drive that many wild animals and other animals in nature also go through. And we were discussing examples of some rat species and praying mantis, right? Where I think Atif told that he has seen some video like this where female mantis is chewing the head off of a male mantis while they are copulating, right? Because the sexual drive overpowers all other drives, or you can say behaviors. Now today we'll be going. Yes, Saifullah has joined. Let's just allow him in. Yes, good evening, Saifullah. Still connecting to audio. Okay. So today we'll be discussing about testes and everything that is happening inside, which means spermatogenesis majorly, and this is a very important part. For this, I already asked you to go back and read about meiosis. I hope everyone has read about meiosis and the stages in detail by now. Yes, just say yes or no. Atif, Mazia, Saifullah, Mohsen, Hania.
Yes, sir.
Yes. Emuna and Minashi.
Yes, sir.
Okay, good. See, if you have not, please do it, because today I'm going to go into the details. I'm going to start this thing. We cannot not start it. We have to start it. Okay, you all can see this diagram, this figure.
Yes, sir.
Something happened. No? Yes. Happened, but somehow everything got select. Okay, it's fine. Get back to normal. Yes, you all can see this diagram.
Yes, sir.
Yes, so this I have taken from this is one reason probably I do not like online classes, but it's fine. Yes, okay. Is it visible now? This is a decent size. Yes, we're back. Namana. So today we'll be discussing about this process of formation of male gametes, which is called spermatogenesis. It looks like a very straightforward, simple process, but it is not. It is very complex. And also, before going into this, we have to understand testes as an organ. So I have already told you, who's writing what? So did you give any notes? Notes regarding, so I have given you notes about the hormonal control yet. I will be starting with the testes now. Okay.
Okay, sir. I was having some technical problems. Sir. That's it.
Okay. So tell me, what do you, when we were discussing sexual reproduction in plants, can someone tell me what was the equivalent of spermatogenesis there quickly, just to put things in context? Minashi, what is the equivalent process of spermatogenesis in sexually reproducing plants? Am I audible?
Yes, sir.
Yes, sir.
Remember plant reproduction? chapter which just before this one, there, what were the male gametes called? Ma'am, male gametes in a plant or equivalent of a male gamete in a plant are called what? Pollen grains. No?
Yes.
Yes. And how are pollen grains formed? Remember? Gametogenesis there?
Microsporogenesis.
Microsporogenesis. Perfect. And it was happening through pollen mother cell, PMC. Remember? And I told you that PMC undergoes meiosis and from one cell makes 4 microspores, correct? Pollen
Yes, sir.
mother cell makes 4 microspores. Yes or no? Yes?
Yes,
Yeah.
Yes, sir.
Here also in spermatogenesis. Now look at this figure here. So this first structure that I'm marking here, primordial germ cell. You know, germ cell is something we call something as germ cell or sex cell, which gives rise to gametes later on. Okay. So germ cell, and you can see here that this germ cell is diploid or haploid. Tell me.
Diploid.
Yes, it is diploid. Okay. Can you see 2 copies inside? This germ cell first, see what is happening here. Before it starts forming gametes, it undergoes mitosis to make more of its kind, which are called spermatogonium. So upon mitosis, it forms spermatogonium, which are again diploid. Do you understand this spermatogonium? So primordial germ cells, can you see here DNA replication happened, which happens in mitosis? And from one cell, 2 cells are formed, each of 2n, which means it is mitosis. And then this, after mitotic division, the primary spermatocyte is formed. Now primary spermatocyte, now as the word suggests, something is called a gonium, where it has the potential to give rise to that thing. Spermatogonium will give rise to sperms, but still has not been committed yet into that process. So it still has not undergone meiosis. Is it clear, everyone? It just has the potential, so it will do at one point of time. When it is committed, it is called a spermatocyte. First, it is called a primary spermatocyte. So this cell here is a primary spermatocyte, which means the word spermato comes from sperm and cyte is cell. So this cell eventually will now become a sperm someday, sometime down the line, it will become a sperm. So first meiotic division happens in primary spermatocyte. Write this down. Meiotic division, first meiotic division happens in primary spermatocyte. Entrance question. First meiotic division means M1. Remember I told you that meiosis as one cycle, as one cell division cycle, it actually has 2 cycles of division happening. You remember this? No one remembers nothing? People, you have to speak. You know how this class works.
Yes, sir.
So the first meiotic division, which is meiotic division 1, happens in a primary spermatocyte, and as a result, there are 2 secondary spermatocytes formed. Okay.
Yes.
Now the meiosis has not commenced yet. It is not over yet. There is meiotic division 2 also yet, right? Because from one cell, ultimately, how many daughter cells have to be formed in meiosis?
4.
4. And meiosis always starts with a diploid cell. Remember? Basics of meiosis.
Yes.
can only do meiosis in a diploid cell. We can never do meiosis in a haploid cell. Do you understand why or why not? If someone has confusion why haploid cells cannot undergo meiosis, please tell me right now. Everyone is clear why haploid cells cannot undergo meiosis?
Yes.
Yes, they can only undergo mitosis. So everything till primary spermatocyte was diploid. Even the first meiotic division, which made two secondary spermatocyte. Now tell me, after the first meiotic division, what is the ploidy? Is there a change in the ploidy level?
Sir, can you repeat what you asked?
For example, I'm saying primary spermatocyte is diploid, right? 2n.
Yes, sir.
That's why it can start meiosis. Only diploid cells can start meiosis, which means primary spermatocyte was diploid. These are all entrance questions. I'm trying to explain you it through a diagram so it stays in your head. So primary spermatocyte got committed into the act of meiosis, which means it was itself diploid. And after the meiosis 1 finished, the first cycle finished, there were two daughter cells, which were called secondary spermatocyte. Now, secondary spermatocyte will be diploid or haploid?
Diploid.
Haploid. Because the meiosis 1 reduces the ploidy. Remember, I also told you meiosis 2 is equivalent to mitosis. It is just like mitosis. No one remembers nothing. What?
Sir, I confused it with meiosis 1. I thought meiosis 1 was equal to mitotic division. That's why I said 2.
Meiosis 2 is equivalent to mitotic division. Yes or no?
Yes, sir.
Can you go back to your notes, everyone, and confirm that you have written the same? Mamuna, what have you written?
Yes, sir.
What have you written? Meiosis 2 is equivalent to mitosis, or meiosis 1 is equivalent to mitosis? Which is an equational division? Meiosis 1 or meiosis 2? And both 1 and 2 together makes meiosis, the complete cycle. So which part, 1 or 2, is equational? Which is reductional, which is equational? So write down M1 is reductional. Meiosis 1 is reductional, whereas meiosis 2 is equational. And meiosis 2 is like mitosis. Okay, everyone? Will this be in your head now? Say yes or no?
Yes.
Yes, I think.
Yes.
Now, if I ask you the questions, which cell starts the meiosis in spermatogenesis? Which cell undergoes meiosis? Primordial germ cell, spermatogonium, primary spermatocyte, or secondary spermatocyte? So this is a legit question. Answer. Which undergoes meiosis? Primordial germ cell undergoes mitosis to make more of its own kind and to make spermatogonium. Okay. They all are diploid. Primary spermatocyte is the first stage or the first cell that undergoes meiotic division, M1. And as a result forms secondary spermatocytes, which are haploid, and then they undergo meiotic division 2, and from 2 they make 4 cells which are called spermatids. Is it clear, everyone? Spermatids
Yes, sir.
are also haploid. So secondary spermatocyte is the first stage in spermatogenesis, which is haploid. Okay, keep writing all these things. Write down primary spermatocyte. Primary spermatocyte undergoes meiotic division 1. Undergoes meiotic division 1. And primary spermatocyte is a diploid cell. And after meiotic division 1, two haploid cells are formed, which are called secondary spermatocytes. So write down. Secondary spermatocyte are the first haploid cells in the process of spermatogenesis. Secondary spermatocyte are the first haploid cells in the process of spermatogenesis. All of this is happening inside the testes. We'll come to the structural or anatomical things later. Let's first understand the cycle. Then,
Are the first haploid cells after that? Can you repeat?
yes, are the first haploid cells in the spermatogenesis process. Everything above that was diploid, right?
Yes, sir.
Now write down after meiotic division 2, after M2 is complete. After M2 is complete, meiotic division 2 is complete, which is equivalent to a mitosis only. This you do not have to write, I'm just telling you. So after M2 completes, 4 haploid spermatid cells are formed. 4 haploid spermatid cells are formed. Now these spermatids are not mature sperms because they do not have a tail. They are just like cells. Okay, any other cell, any other haploid cell. They do not have a structure, head, a tail structure which a mature sperm has. So for sperms to become mature, these spermatids needs to develop tail. Okay, is it clear? And a head region, acrosome. In this process of starting the making of head and tail happens in the lumen. Now, where it happens, let's come to that. So, is this graph clear to you, everyone? Everything starts with a diploid cell called primordial germ cell. These cells forms the gamete and the gonads. Gonads are the testes and ovaries. So everything related to the sexual structures or the sexual organs comes from this primordial germ cell. This is known as PGC. Some books and some entrances write it as PGCs. Okay. They make spermatogonium cells after mitotic division. Then the spermatogonium, this is the mitotic division. Spermatogonium makes primary spermatocyte, which is still a diploid cell, but this first undergoes meiosis. Atif is asking, sir, does primordial germ cell divide twice to form 4 spermatogonia? Spermatogonia, no. There is only, so primordial germ cell, if it is undergoing mitosis, it will only form 2. So one primordial germ cell can only form 2 spermatogonia. You can see it here. Primordial germ cell, when it forms spermatogonia, from 1 it is 2. Can you see here, Atif? Is the doubt clear?
Yes,
From
sir.
1 primordial germ cells, you cannot make 4 spermatogonia. You can only make 2 spermatogonium cells. Is it clear, Atif?
Yes, sir.
Yes, because it's mitosis happening. So it has to go. So from these two spermatogonium cells, each spermatogonium can become primary spermatocyte. It is just a changed form of the spermatogonium only. The difference only is that once any spermatogonium becomes primary spermatocyte, it is committed to the act and to the process of becoming a sperm. Do you understand? The difference is like this. So till you are in school, okay? See, you have the potential to become a doctor. You are in school in class 10th. You are a primordial germ cell. You can either become gonad, a vet, a doctor, biotechnologist, anything related to biology, right? So primordial germ cell can also give rise to the gonads and the gametes. Is it clear? Say yes or no.
Yes.
When it
Yes,
becomes
sir.
spermatogonium, it is a little more specialized. Like this is spermatogonium is when you come to 12th. Now you will be sitting for entrances, and if you clear the entrance, you become the primary spermatocyte in your first year of MBBS. What an analogy, but it just came to my mind. So once you are in the first year of your MBBS, you are committed to becoming a doctor now. Yes or no?
Yes.
Yes, sir.
So technically, there is no way that you are in the first year of MBBS and when you ended with your studies, you became an engineer. It cannot happen. You will only become a doctor. So once something is a primary spermatocyte, it will only now become a sperm someday. Is it clear? Secondary spermatocyte is more specialized because now meiotic division first has happened and these are haploid, the first haploid cells, which means you are in the second or third year where you now started doing the practical or surgical experiments on the cadavers, the dead bodies. Now there is no way, no turning back. And once you take, like you finish your studies, you are spermatids, still not completely mature to become a doctor. So you need to go through 1 year of compulsory internship. Yes or no?
Yes, sir.
Yes, in that 1 year of complete internship, you become a mature doctor. So this is where spermatids go for their internship, develop tail, acrosome, they get structural modifications and become a mature sperm. Now ready to do what they have to do, fertilize the ovum. Is it clear, everyone? Any doubts?
No, sir.
No? Okay, very good. Now let's come to this image here. Let me zoom it a little. I can, yes. You all can see clearly? You all can see the screen clearly?
Yes, sir.
Now look, this is the structure of a testis, okay? This is one testis. Do you see these tube-like structures in this?
Yes, sir.
These tube-like structures are called seminiferous tubules. Inside these seminiferous tubules, now they have just taken one of these tube and they have shown a image, like a zoomed in image, and then this part of this picture is shown here. So basically this is a section of a seminiferous tubule. Everything is happening inside these seminiferous tubules, okay? So testes, and what is this structure here? This is called Retted Testis. In last class, we in the NCERT diagram, I told you this. Let's go a little up and try to relate. You see this here? The same thing is shown here. So there are the testicular lobules are basically the seminiferous tubules are present here. Is it clear, everyone?
Yes, sir.
Then this part which you are seeing, which is marked as Retted Testis, is this part here, okay? And then from there you go into this epididymis. This also has three parts: the head, body, head, abdomen, and the tail. Head part is this, the abdomen, and this is the tail. And where does this go? This is going to vas deferens. This structure. Let me go up. Look at this. So this is the epididymis, and then it is coming towards this out. This structure is vas deferens. Okay, it's marked here, but this is it. Is it clear? The anatomy is clear, right?
Yes, sir.
Now look at, focus at this image here. So you see towards the wall, everything is happening. So this is the outermost wall. This is the wall of seminiferous tubule. So the structure is like this. In each testicular lobule, write down this line. Inside the testis, in each testicular lobule, there are 1 to 3 or 4 seminiferous tubules. There are 1 to 4 seminiferous tubules. And inside the seminiferous tubule, there are 2 types of cells. First is called the Sertoli cell. So write down this cell here. Write down. Can you see this? Sertoli. So one type of cell is the Sertoli cells. Write down. Inside the highly coiled seminiferous tubule, inside the highly coiled seminiferous tubule, there are randomly scattered Sertoli cells. Okay.
So can you repeat the point once more?
Yes. Inside the highly coiled seminiferous tubules, there are randomly scattered Sertoli cells. Correct? This is what I said. Now write down the function of Sertoli cell. This is asked. First thing is Sertoli cells. So they are randomly scattered. Their function is to support and nourish the immature sperm cells. To support and nourish, which means to provide nutrition, basically. To support and nourish the immature sperm cells by providing nutrients Now look here, you see that in this image? So apart from Sertoli cells, there are these spermatogonium. You see these spermatogonium? They are towards the outer wall, right? Towards the wall. And as they progress more and more, so as they become more and more a gamete, they start moving inwards towards the lumen. Now lumen is this structure inside a tube. Inside the seminiferous tube, it is hollow at the center, which is called lumen. Okay, and towards the edges, the hierarchy goes like this. Spermatogonium cells are at the top, which were formed from primordial germ cells, which will be even like further. So spermatogonium, then they become primary spermatocyte, they come a little down towards the lumen. Then when they become secondary spermatocyte, they come a little more down. And when they become spermatids, they are a little more down. And then when they become mature sperms, they move out through this lumen. Is it clear, everyone? So it's an outside in journey from the seminiferous tubule wall towards the lumen. Okay. Maturity, as the cell matures towards becoming a sperm, it keeps going towards the lumen. Now in this movement, neither spermatogonium nor the primary spermatocyte nor the secondary spermatocyte, they did not have tail, right? Yes or no? I can understand that a mature sperm can move on its own because it has a tail now. Till the time it was not having tail, how was it moving down towards the lumen? Any idea? Which is the cell which is the caretaker cell till they become sperm? Sertoli cells. Is there any confusion, or people are not speaking by will? Is it clear that Sertoli cells are the caretaker cells of?
Yes, sir.
Okay. So whatever will happen to the spermatogonium, primary spermatocyte, secondary spermatocyte, and spermatids, it will happen because of Sertoli cells. So Sertoli cells does not only nourish these cells, but also transport them from the periphery towards the lumen. So write down. Sertoli cells help to transport immature sperm cells. When I'm saying the word immature sperm cells, it is applicable on spermatid, secondary spermatocyte both. Okay. Is it clear, everyone?
There had connection problems. Can you repeat the point?
Sorry, what, Atif?
There I had some connection problems. Can you repeat the point?
Yes. The point which I said before, or which point?
Yes, sir. On about Sertoli cells.
All about Sertoli cells.
No, sir. The last point that you said.
Okay. So I was saying that Sertoli cells helps to transport the immature sperm cells. And I was telling you, when I say immature sperm cells, it means spermatids. Okay. You can also
Yes, sir.
include secondary spermatocyte, but majorly it means spermatids. They are the immature sperm cells. So transport of immature sperm cells from the outer surface of the tubule, from the outer surface of the seminiferous tubule towards the central lumen. So till they are not capable of moving themselves, Sertoli cells also helps in their transport. Okay. Now, if you understand the process, the testes or these seminiferous tubules, are constantly producing sperms all the time, 24 hours a day, 365 days a year in a male after puberty. Okay? So if you take the cross section of any seminiferous tubule from any adult male at any given time, you will see all the stages simultaneously in that seminiferous tubule. Yes or no? Yes or no? Do you understand what I just said?
Can you repeat it again, sir?
I was saying that it is a continuous factory. Spermatogenesis is happening every minute, every second, all the time in a adult male. Is this clear?
Yes, sir.
So if I take any adult male's testes and take out a seminiferous tubule to examine from it, make a cross section of that seminiferous tubule and mark the stages of the cell, I will find all the stages at every given point, right? In that picture. So there will be spermatogonium also, there will be primary spermatocyte, secondary spermatocyte, some spermatids, and also some mature sperm all the time, correct? Because it's a continuous process. Now, is it clear what I just said?
Yes, sir.
So write down. Sperm production is a continuous process. Sperm production is a continuous process in a adult, in a mature testis. In a mature testis. And one germ cell starting from the very beginning here. Look here, one germ cell at the beginning of this cycle. One germ cell takes around 2 to 3 months. 2 to 3 months. It's a range. Okay, it's around 60 to 80 days. You can write in terms of days. It takes around 60 to 80 days. Many books write they take 75 as a median, 75 or 74 days, which is like 2 months and 15 more days, correct? So between 60 to 80 days every germ cell takes to reach final maturation as a sperm. To reach final maturation as a sperm. So it's a very long process. And one germ cell takes around 2 months and some odd days, around 10 weeks, if I'm not, yes, around 10 weeks to become a mature sperm. So in terms of week, it is 8 to 10 weeks. In terms of months, it is 2 to 2.5 months, and then days accordingly to become one mature sperm. But millions of them together start their journey from the germ cell onward. So millions of them actually every day are getting mature as mature sperms. Is it clear, everyone? And write down. There are many resting stages. There are many resting stages where the cell will rest for some time in that 70-75 days. There are many resting stages in between. But there is no as such a long arrest. There are many small resting stages, but spermatogenesis is different from oogenesis Because remember I told you, when does oogenesis starts in a female? Tell me.
In the embryonic development stage.
Yes, and I also told you that when it reaches anaphase 2, it gets arrested, right?
Yes, sir.
So it completes meiosis 1, and when it reaches a stage, it gets arrested. And it stays arrested for very long period. After birth till puberty, it stays arrested in that stage only. After that, every oocyte starts from that stage and become mature. So there was a very long pause in between, which is not seen in spermatogenesis. There are many small intermittent resting stages, but they are not big arrest or pauses. Is it clear, everyone? How is oogenesis different from spermatogenesis?
Yes, sir.
Yes. Now write down. The secondary spermatocyte, which is the first haploid cell, remember, before spermatids. So which is the closest in some books, like I think also in NCRT, they write sperm as secondary spermatocyte. So secondary spermatocyte is a sperm without a tail, but you know that it's still not mature. It is the first haploid, so it is haploid like a sperm, but still it has to go a very long way before it can fertilize. It can get the capability to fertilize an ovum. So write down, the secondary sperm cell is a haploid cell like the mature sperm.
Can you repeat from the start?
Yes, the secondary spermatocyte or the secondary sperm cell. If I say spermatocyte or sperm cell, it is the same thing, right? Spermatocyte also means sperm cell. Cyte word is for cell. Is it clear?
Yes, sir.
Secondary sperm cell or secondary spermatocyte is haploid like a mature sperm, but still has to go many modifications, still has to undergo many important or vital modifications before it can fertilize an egg. Before it can fertilize an egg. Is it clear?
Yes, sir.
Yes. So Sertoli cell is clear to you. Spermatogonium is clear. Primary spermatocyte is clear. Secondary spermatocyte is clear. Right?
Yes, sir.
Now write down maturation. Maturation of sperm. So before this stage, till spermatids, it is just like any cell, right? Any cell of the body, which is like of roughly a oval shape, no tail, no head, no distinguishing structures, no movement ability, like that. So when a sperm mature, what all things happen? So first point, write down. First point. Maturation of sperm. Maturation of sperm brings certain changes. Brings certain changes in the shape and form of the sperm cell. Yes. bring certain changes in the shape and form of the sperm cell. Now start writing bullet points. I will be telling you what you have to write if a question comes in board about maturation of sperm.
So changes in
Changes in, Minashi, changes in?
Change in the shape and form of the
Changes in the shape and form of the sperm cells. Shape and form of the sperm cell. Now start writing as bullets. First point, see, look here. When it was a cell, roughly a oval normal cell, versus when it will become a head. So a head needs to have a shape, right? A pointed shape with a top cap of acrosome at the top. Yes or no?
Yes, sir.
And sperm has to become more toned and more compact, right? Compact as a structure, so that it does not take much of space and resistance when it is flowing in the fluid, right? So it's
Yes.
the structure of sperms becomes more and more streamlined with a tail. Okay, so first thing that happens is the nuclear material, write down the nucleus, first bullet, the nucleus becomes more condensed and oval in shape, because oval shape is more streamlined than circular shape. You all understand what is streamlined? Saifullah, what do I mean by streamlined shape? Minashi, you understand what is streamlined?
that mitochondria line.
Mitochondria like.
No, that the lines that are present on the neck.
No, but what is streamlined shape? Streamlined shape is what? Anyone? Someone is writing. Mazia is writing shape of boat or a fish. Any idea? Who would like to? Mamuna, you understand what Mazia said? Streamlined shape. She's right. She gave examples that shape of a boat or a fish is streamlined. Streamlined shape is those shapes which do not get resistance from the medium in which they are flowing or moving or flying or swimming. Is it clear? Is it clear?
yes, sir.
The fastest fish are the most streamlined ones. Okay. So if you see someone has to just go speedily in the water, suppose you are diving in water and you want to go very speedily inside, you just stretch your hands and make your body streamlined. Yes or no? Same is for diving. Someone is skydiving. Or have you seen birds that predate on fish? Some birds, they keep coming from the sky flying and then go dive inside the river and take a fish out. Anyone has seen those predatory fish, predatory birds?
Yes, sir.
So when they dive in the water, do they have their open wings and they are diving with their wings open?
No.
No, they close it and they make their body more streamlined. So this is streamlined. A perfect streamline. So when air starts or water starts coming towards it, it will get split very easily. And it will have very less resistance. If this is the shape of a body and it is moving in water, it will get a lot of resistance, right? Very difficult to swim with this shape. Yes or no?
Yes, sir.
So this is a streamlined shape. So what I was telling you, please someone can repeat the line, Minashi, the last line you wrote.
The nucleus becomes more condensed and streamlined.
Yes, the nucleus becomes more condensed and oval in shape, right? I said oval in shape. Oval shape is more streamlined. Yes. Write down. This oval shape becomes the head of the sperm. This oval shape becomes the head of the sperm. Okay. Next point. The head is covered by a cap-like structure called acrosome. Acrosome.
The head, sir.
The head is covered by a cap-like structure. Cap-like structure. It's more like a skull cap over the top of the, so our heads are oval in shape, and if you put a skull cap on top of it, it will resemble, or it will be analogous to a acrosome. Okay, cap like structure called acrosome. Now write down, what does write down this question. It is a board's question. What is the importance of acrosome in a sperm head? If there is no acrosome on the sperm head, that sperm is immature, and that sperm can never fertilize a ovum naturally. Why I said naturally? Because artificially we can fertilize ovum taking any sperm we want. So write down, what is the importance of acrosome as a structure? The answer is acrosome plays a vital role. How many of you remember what used to happen when pollen grains used to fall on the stigma? Anyone? What was the next step? People.
Pollen tube is generated.
So any pollen that falls on the stigma will just generate a tube?
A pollen pistil interaction
Yes. Why was that interaction important?
to recognize the correct pollen grain.
To recognize the correct species of that pollen, right? If it is of the same species. And it was a biochemical interaction, correct?
Yes,
If
sir.
that interaction will not happen, there will be no entry of that pollen tube inside the stigma and style, correct?
Yes.
So do you think we need a similar kind of mechanism here also? Because sperms are motile, and if that sperm reaches the ovum, it first has to ensure that the sperm is of the same species, so that the ovum can allow it inside and let it fuse with its own nucleus and become diploid zygote. Yes, there is an importance, right? Yes.
Yes.
So write down, acrosome is important during the sperm ovum interaction. During the sperm ovum biochemical interaction. To gain entry into the egg. To gain entry into the egg. That's why acrosome is at the top. So when a sperm is moving, so if you see this pen as a sperm, so this cap is acrosome. Okay, this cap. So the first thing that will touch the surface of the egg is the acrosome. Another question: which structure first touches the surface of the egg? That is acrosome. Okay, because acrosome contains certain chemicals and also some enzymes. Now I will tell you why those enzymes are important. So I will tell you this later on. Now let me tell you here only. So a sperm, the structure of sperm is clear to you, right? I'm just giving you a spoiler of the female reproductive system here only, giving you the structure of a rough structure of the ovum. So ovum has a plasma membrane, right? Because it's a cell, so it will have a cell membrane. But this is not the outermost layer that is exposed. Above that plasma membrane, there are many cells which cover, and there are many layers which cover this ovum inside. Corona radiata and other follicle-like structures. Okay, someone used the word follicle, right? So when a sperm comes in, it is this. Like this is again not to scale, but it is very small. This is why sperm is known as microgamete, because it's the word micro means small. Anyone there? Anyone alive? People. Sperm is microgamete. Yes. And ovum is macrogamete. Because humans are heterogametic. Remember, heterogametes, different type of gametes in structure and size, etc. So when this sperm first tries to reach the ovum, it has to first encounter a lot of layers, protective layers, before getting inside, no? And this very small, tiny sperm does not have that force or capability to just physically remove those layers by force. So at the tip of the sperm, inside these acrosomes, there are enzymes that helps in degrading these protective layers so that it can reach the surface of the ovum. Is it clear?
Yes, sir.
And one of those enzymes which is present in the acrosome it's called hydrolytic enzymes, which break down things. And one of that hydrolytic enzyme is hyaluronidase. Asked in NEET. Write it down. Hyaluronidase is an enzyme present in the acrosome of the sperm, which is a hydrolytic enzyme and helps in reaching, like degrading the protective layers around the ovum and reaching the surface of the ovum so that it can fertilize and get inside. When it reaches the surface, again, there is a interaction, a biochemical interaction that happens. A polarity change happens between the membrane of the ovum and the membrane of the sperm. The moment sperm touches the membrane of ovum, there is a polarity reversal, a phenomena which we call polarity reversal, and the sperm which has touched the ribbon, it's like a race, no? So there is a ribbon at the end of the race. The person who comes first breaks that ribbon. So the first sperm that touches the ovum, the head of that sperm reaches inside and the tail stays outside. Only the head goes inside. Not even the tail is allowed because tail is a waste material. Now, tail does not provide anything genetically to the ovum. So ovum will not even allow tail to go inside. Only the head part goes inside, which contains the nucleus, the one copy of genome for which the ovum needs the sperm. And the moment this happens, no matter how many more sperms come and touch the surface, no sperm can enter inside. So the gates are shut and the membrane will not allow, the membrane will become repulsive to any other sperm and will not allow any sperm to enter. Can someone tell me what is the magnificent brilliance behind this phenomena? Why is this important? What would have happened if two sperms simultaneously could have fertilized the same ovum? What will happen? People. Someone is writing in chat. Twins, wrong. Twins are not formed when two sperms fertilize one ovum. That is a very wrong misconception we all have. And as biology students, you have to get rid of that. This is why I'm asking the question. If two sperms are allowed to enter the same ovum, what will happen? Write it as a homework. Write it as a in-class assignment. Question is, why two sperms are not allowed to enter one ovum during fertilization? This whole brilliant mechanism has been evolved and put in place just to make sure that the first sperm that touches the surface gets inside and rest all others are just not allowed. So no other sperm can enter inside. Only one sperm can enter, and one egg nucleus can fuse with one sperm nucleus, and both haploid will become diploid now. So why? What will happen if this happens? This you have to write one small paragraph, and I will ask everyone's answer in the next class. I've just told you this is not how twins are formed. We'll come to twins. It's a very interesting phenomenon also. But acrosome is written, everyone. Shall I move forward? Everyone has noted down the homework.
Sure.
Yes, Minashi? You're saying something?
No, I said it.
Okay, you were saying yes. Okay, write down. So acrosome has to be there. Next point. Towards the opposite end of the head, a tail piece is developed. A tail piece is developed. A tail piece is developed. And how does a tail, where does tail come from? So there are two ways to put a tail on a round thing. So let's say my fist is a cell. Now one thing to make a cell, make a tail, I take something which I can attach to the cell and it will be like a tail. Yes, something externally attached, which is not a continuous part of the cell, like flagella. It's like a motor-like thing attached, right? But
Yes, sir.
but in case of sperms, the tail is made from the cytoplasm of the secondary spermatocyte, secondary sperm cell. So write down, the tail is derived from. This is a question. All of these points that I'm telling you are actually questions, because this is a very, you have to understand this process critically. Every point can be asked. How is tail formed? Which structure is responsible for a sperm tail? So write down, the tail of the sperm is derived from the secondary spermatocytes cytoplasm. Okay. I have already told you about the neck piece of the sperm. Like the neck piece is lined by around 50 to 75 mitochondria, right? You all have written that to provide energy to the tail. Yes or no? Just above. Yes, here. So about this neck part, I've told you already. So I'm skipping the neck part. The tail is clear. It is derived from the secondary spermatocytes cytoplasm only. It is not something like a filament that comes from external, like that can be attached. So this is about maturation. Write down. After the maturation, Write down. After the maturation. So what happens now? Look at this figure. Now this here, the last one here, it looks like a very mature sperm. This one. It has an acrosome, it has a neck clearly visible, it has a very good tail. So what happens now? And it is towards the lumen also, right? This is the lumen. So it is towards here. So it can move out from the seminiferous tubule, right? And when it will move out from the seminiferous tubule, what is the trajectory that a sperm will follow has also been asked in question. So write down. From seminiferous tubule, it goes into the retted testis. Goes into the retted testis, and then you see these structures here, these ones. Everyone can see these structures, which I'm marking right now. These tube-like structures. So this bigger one is called vas deferens when it goes out, up. But these smaller ones between the epididymis and the retted testis is called vasa efferentia. What is called? There is something called vasa deferentia and vasa efferentia. Okay. So from retted testis it goes into vasa efferentia and then into epididymis. Is it clear, people? After maturation, seminiferous tubules into retted testis, from retted testis, vasa efferentia, into epididymis. Okay. Say yes or no? Now we will study about epididymis. Why is that important? So write down. When the sperm reaches epididymis, actually, how should I explain this? So they are mature, okay, but still there are certain things, some fine tunings that needs to be done to that sperm, to that mature form of the sperm, so it becomes, it gets its motility. You understand? It's like when someone starts walking, so before that kid or that child starts walking, all these structures are completely formed, right? Legs, hands, bones, everything is there. So it's a mature structure which is now able to walk. Correct? Yes or no? Say yes or no. People, are you there?
Can you say again?
I was saying, just like when a child starts walking, so before it starts walking, it is all the structures that are required to walk are in place, right?
Yes.
That is called a matured anatomy. So the kid has bones, legs, tissues, muscles, everything. But the moment the kid takes the first steps, are these first steps as balanced and like, are they very fast and balanced? No, right? It has to go through some few days and months of walking practice so that it can walk properly, right? And over the time, other structures of leg, tissues, bones also keeps on getting mature. So they are already formed, but they still need fine tuning. Similar thing happens in epididymis. So write down. Epididymis is a crescent shaped structure. Crescent shaped. So if you can see it here clearly, see this is the testis and this is a crescent shape, right? Can you see this? It's a crescent shaped structure sitting on the top of the testis. Sitting at the top of the testis. Sperm cells are matured and stored. Sperm cells are matured in terms of motility. Write down in terms of motility. Structure wise, they are already mature. So no new structure is added in epididymis. All these structures like tail, neck, acrosome, head has already been added in the seminiferous tubule. In the epididymis, the maturation is more from its motility perspective. Do you all understand this? Is it clear? No confusion, no doubt?
Yes.
Yes. So write down. It is matured with respect to its motility and is stored there. And is stored there. Now, epididymis has three parts: the head, body, and tail. This is the parts of the epididymis as a structure: head, body, and tail. Now, to go into details of this head, body, and tail and what happens, let's start it in the next class because I think it's time. I will not be able to finish if I start this. But just know a few things. Write down. The smallest region is the tail. So this was one question. So I have marked it. Tail is the smallest region. In epididymis, whereas in a sperm, tail is significantly larger as compared to head. Is it clear? If we talk about tail of sperms, it has a significant large tail as compared to the head and the neck. Whereas if we talk about the structure of epididymis, which is an organ, tail is the smallest region. Okay, so don't get confused here. Okay, so I'll stop here on epididymis today. About testis. Anyone has any doubt about this whole process? Please ask me. So I give you 2 minutes before you go. Those who have done, if you need to note anything from this figure, or if you note, I would appreciate if you all make this flow chart. Everyone has made this flow chart in their copies. Yes or no?
Yes, sir.
Yes. So those who have made everything can leave, and I'll see you in the next class. If someone needs to note anything down from this figure, can do this. Okay, it's there for the next 1 or 2 minutes. If you have anything to ask, please ask me any doubts or something. Otherwise, this is the end. I'll see you in the next class, people. Bye bye.
Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Saifullah and Hania, are you still noting something now?
Sir, I'm done. Thank you.
Okay. Saifullah?
I'm doing this flow chart, sir.
Okay, do it. Take your time. No problem. Let me know when you're done, okay?
Okay, sir.
You're done, Saifullah?
1 minute, sir.
In the meanwhile, hello, Samara. Am I audible and visible to you?
Yes, sir.
So, this is your first class, correct?
Yes, sir.
Yeah, okay, just give me and Saifullah some time, okay? He's just noting a few things down.
Done, sir. Thank
Okay,
you, sir.
Saifullah, see you in the next